Bismillahirrahmanirrahim. Welcome. This is Biochemistry 2 course called Chem 342, and today we are having lecture 30. Our today's topic is vitamins K. This is your teacher Dr. Naeem Khan, Chemistry Department, Cust. Outlines for today's topic include: we will discuss the chemistry of vitamins K, and then we will highlight the different metabolic process through which vitamins K passes in human body and take vitamins K in the food. Then we will highlight the important sources of vitamin K, and we will point out the requirements of vitamins K for human beings. And at the end, we will discuss the important functions of vitamins K. To have a start, chemistry of vitamins K. Vitamins K are also called as anti-hemorrhagic factor, or sometimes it is called coagulation vitamins. Why? The reason is because vitamins K have very important function in the blood clotting. So they have a unique power of the blood clotting, and that's why they are called as coagulation vitamins or sometimes anti-hemorrhagic factors. Naturally, there are two forms of vitamin K known, and they are designated as vitamin K1 and K2. Uh, Vitamin K1 was first isolated from alfalfa in 1939 by Dave et al. Dave and his research associate for the first time isolated vitamin K1 from alfalfa in 1931, while vitamin K2 was isolated from fish meal by Dozy and his co-workers in 1939 too. The relative biological potencies of vitamin K and K1 and K2 includes: if K1 potency is 100, then vitamin K2 is 80. So the potency of K2 is less than K1. In the introduction, vitamins K, they have very important role in the blood coagulation. So that's why it is known as coagulation factor or coagulation vitamins. It is also known as anti-hemorrhagic factors. Naturally, two forms of vitamin K have been isolated, and these are designated by K1 and K2. K1 and K2 both are isolated in 1939. K1 from alfalfa by David Hall, while K2 was isolated by from fish meal by Dozy and his co-workers. Potencies of K1 and K2 are: vitamin K1 is 100, so vitamin K1 is considered as the exact potency or 100% potency of K, while K2 is 80%. Chemically, from the chemistry point of view, K1 and K2 are basically quinones. They are derivatives of quinone. Their basic structure is quinone. Vitamin K1 and K2 differ from each other in the composition of side chain present at carbon number 3 of naphtha-quinone ring. Actually, both K1 and K2 have naphtha-quinone ring in their structure, and there is a side chain at carbon number 3. So, naphtha-quinone ring is the same in both K1 and K2, but the two forms are different from one another on the basis of the side chain that is attached at carbon number 1. So, they are quinone derivatives having naphtha quinone ring in their structure and having a side chain at carbon number 3, which is different in K1 and K2. There is a phytol radical in K1 and vitamin K1. Vitamin K1 is having phytol radical, while this farnesyl radical is present in K2 at carbon number 3. K1 is C31H46O2, while K2 is C41H56NO2. So both are quinones having naphtha quinone ring in their structure, and they have side chain at carbon number 3, which is phytol in case of Q1, and this difarnesyl in case of K2. Vitamin K1 is chiefly found in plants, and it structurally has 4 isoprene units in its side chain with double bond in the first unit only. If you look into this structure, this is K1 structure, this is quinone, naphtha quinone structure, and it is having a side chain at carbon number 3. And this carbon number 3 side chain is made up of 4 isoprene units, 1, 2, 3, 4. And these isoprene units having a double bond in the first unit only. This is number 1, and these are 3. So total are 4 isoprene units, but the first isoprene unit is having the double bond. Vitamin K1 is basically found in plants, and it contains 4 isoprene units at carbon number 3 side chain of the naphtha quinone, and the first isoprene unit is having double bond. Coming to K2, this is chiefly found in animals and contains 6, not 4, 6 isoprene units, and its side chain each with a double bond. Each side chain is each units in the isoprene contain a double bond. Look at this. This is a structure of K2. Naphtha quinone is the same. At carbon number 3, we have side chain, and here we have 6. This is 5, and this is 6 naphtha isoprene units, and each of the isoprene units is having double bond. Here in K1, we were having double bond only in the first isoprene units, but in K2, we have double bond on each isoprene units. And second difference is here we have 4 isoprene units, but here we have 6 isoprene units. So basically K1 and K2 are quinone derivatives, naphtha quinone in their structure. Carbon number 3 is having a side chain, and on the basis of the carbon number 3 side chain here, we have um, the differences among the K1 and K2. So K1 is having 4 isoprene units, while K2 is having 6 isoprene units. There are analogues of naphtha quinones which have characteristic like vitamin K, and this is because of their structural resemblance. And very common analogues of vitamin K include menadione and thiacol. We have two derivatives, two analogues for the vitamin K, and these analogues show similar characteristics as that are known for vitamin K, and this characteristic resemblance is because of the structural resemblance. They have activity resemblance the same as that of vitamin K, but this resemblance is because of their closeness in their structure. Look at this. This menadione, which is also known as vitamin K3, and thiocol, which is also having basic structure if leftoquinone derivative structure like this. Because of this basic structure similarity, its activities known to be the same as that of vitamin K. Especially this menadione is known as vitamin K3. Look at this. Menadione is also referred to as K3, having twice as potent as vitamin K1. twice as potent. This structural resemblance is important for the potencies of vitamin K3. This structure can be traced in the GLJ and biochemistry book. Coming to the metabolism, vitamins K is, we know they are fat soluble vitamins, so they are absorbed only in the presence of bile. They cannot be directly absorbed. So whenever bile is available, they are absorbed along with the bile. Absorption occurs in the upper portion of the small intestine, and when bile is there, it gets attached with the bile and is help in the absorption. Avitaminosis. The deficiency of vitamin A may occur when there is a bile is where bile is prevented from entering the intestinal tract. Yes, because we know that for vitamin K we need the for vitamin K, we need bile for absorption. So whenever bile is prevented to enter the intestinal tract, then A vitaminosis will occur, means the deficiency of vitamin A K may occur. Sources of vitamin K. Vitamin K is available in green vegetables such as spinach, alfalfa, or cabbage. These are the good sources of vitamin K. Fruits and cereals, they are not that much known as sources of vitamin K. So they are poor sources of vitamin K. Vitamin K2 is found in meat, dairy products, eggs, and especially Japanese natto, which is made of fermented soybeans and in some intestinal bacteria. Vitamin K2 has a special sources, and that is in the meat, dairy products, eggs, and Japanese natto that is containing fermented soybean. Also, intestinal bacteria contains vitamin K2. A rich source of K2 is putrefied rotten fish meal. This is usually considered worldwide as the richest source of vitamin K2, rotten fish meal, putrefied. Some food sources of vitamin K, uh, for example, if 10 sprigs of parsley contains 90 micrograms. If we take 10 sprigs of parsley, it will contain 90 micrograms of vitamin K. Similarly, a half cup of frozen and boiled collard greens contain 530 micrograms of vitamin K. 1 cup of raw spinach contain 145 microgram of vitamin K. 1 tablespoon if we take of soybean oil, it contains 25 microgram of vitamin K. A half cup serving of grapes contain 11 microgram of vitamin K. So if we take half cup of grapes, it contains 11 microgram of vitamin K. Similarly, a hard-boiled eggs contain 4 microgram of vitamin K. So these are the some important sources of vitamin K we must take, and these are including the parsley, which are containing a high concentration of vitamin K, and also frozen boiled collard greens. And similarly, we can use raw spinach containing vitamin K, soybean oil, and grapes, they are containing vitamin K, and hard boiled eggs. Requirements. Vitamin K deficiency is very rare in higher animals, and this is because vitamin K is provided by food, and also it is synthesized by the intestinal bacteria. As it is synthesized by the intestinal bacteria, so normally animals do not need, higher animals do not need vitamin K, and our food is usually containing enough vitamin K. Also, as we have bacteria in our intestine, so these bacteria are producing vitamin K. So under normal circumstances, higher animals do not need vitamin K. And due to this reason that animals do not need vitamin K, therefore no standard requirement has been set. There is no standard requirement set for the vitamin K, as we are normally not having any sort of deficiency of vitamin K. No tolerable upper limit has been determined for vitamin K. There is no tolerable, normally their concentration is enough, and that's not very high as well. So no upper limit, no standard recommended limit. Normally their concentration in the food or the bacterial production in the body is enough to provide the blood requirement of vitamin K. Toxicity to this vitamin is very rare and it's normally not occur from eating of food. So if you are taking normal food, it will not cause any dangerous effects. So normally there is a safety from this vitamin, and we must not be that much worried about. Functions of vitamin K. Vitamin K important function include vitamin K play an important role in the biosynthesis of prothrombin. And we know prothrombin is a very important plasma protein, and that is required for the blood clotting, and it is produced in the liver. At the start, we said that vitamin K is known as clotting vitamin or anti-hemorrhagic vitamin, and we said that it is having a very special role in the blood clotting. So here, and its this role in the blood clotting is because it is important for the biosynthesis of prothrombin, and prothrombin is required for the blood clotting. How? Look at to this. Vitamin K is produced in the liver, and when it passes here, it produces prothrombin. Thromboplastin calcium presents prothrombin, we know, is then converted to thrombin, and this is having thrombin is required for the fibrinogen conversion to fibrin, and then fibrin actually aggregates along with salt and other important some blood cells and other particles, and this is finally cross-linked and forming actually clot. So the vitamin K role is very important. Vitamin K is providing prothrombin, and prothrombin is when converted to thrombin. This thrombin is having enzymatic activity for fibrinogen. Fibrinogen is another plasma protein, and fibrinogen is converted to fibrin. Actually, the clot is made up of fibrin when it aggregates the small particle, blood cell, and other protein, and this making in the form of mesh, and then cross-linking results in the formation of clot. So this clot will not be formed if thrombin is not available, and thrombin is formed from prothrombin, and prothrombin is formed by the vitamin K. So vitamin K is very, very, very important for the blood clotting of humans. This sketch can be traced in J.L. Jain Biochemistry book. Vitamin K also have a very important role in the electron transport chain and oxidative phosphorylation in mitochondria. Electron transport chain is very important because here energy of the electron is used for the phosphorylation of ADP to ATP, and ATP is the currency of the body. That's very important. This providing energy for all the physiological processes. Vitamin A supports immune system, that is by supporting the growth and distribution of T cells, of white blood cell that protects body from. So vitamin A is very much important because it is having a very important function of the immune system. It protects the body from infection, and this is because they are supporting the growth and distribution of T cell. T cells is a very important cell because it is along with the along with the antibodies. T cells are the second mechanism here, which is protecting the body from the infection. So vitamin A actually supporting the growth and distribution of T cells, which protect the body from infections. Vitamin K helps in the maintaining blood pressure, lower preventing mineralization, keep it constant because lower the blood pressure by preventing mineralization where minerals build up in the arteries. This enables the heart to pump blood freely through the body. Dear student, once again, Winston Todd and GLJN books are recommended, and I would suggest to please thoroughly go to these books, thoroughly study all the points we discussed today, and try to develop a more elaborated and expanded form of these pages, and have developed a thorough concept. And if you have any question, we may discuss next time. From today, I would say thank you, goodbye, Allah Hafiz.